In Chapter 11 of your textbook, we'll cover labeled immunoassays. The topics we'll discuss include formats for labeled assays, heterogeneous versus homogeneous assays, radioimmunoassay, enzyme immunoassays, fluorescent immunoassays, and chemiluminescent assays. Unlabeled immunoassays such as precipitation and agglutination reactions that we discussed in Chapter 10 are very simple techniques to perform, and they require little in the way of equipment. Even though these are simple techniques, they're very insensitive because they rely on a high enough concentration of the unknown to visualize the reaction. In contrast, labeled immunoassays are designed for antigens and antibodies that may be small in size or present in very low concentrations. The presence of antigens or antibodies is determined indirectly by using a labeled reactant to detect whether or not specific binding has taken place. Labeled immunoassays can be used to measure analytes such as bacterial antigens, hormones, drugs, tumor markers, and specific immunoglobulins. There are two major formats for all labeled assays: competitive and non-competitive. Current techniques include the use of fluorescent, radioactive, chemiluminescent, and enzyme labels. In competitive immunoassay, all reactants are mixed together at the same time. Labeled antigen then competes with unlabeled patient antigen for a limited number of antibody binding sites. The concentration of the labeled analyte is in excess, so all binding sites on the antibody will be occupied. After separation, the amount of bound label is measured and used to determine the amount of patient antigen present. In the competitive immunoassay, the amount of bound label is inversely proportional to the concentration of the labeled antigen. This figure depicts an example of a competitive immunoassay. So we have two samples, sample A and sample B on the right. So the unknown concentration of an analyte in a patient sample, noted as the red dots in sample B, compete with labeled analyte, the yellow stars. Sample A is the negative control, so you see there's no analyte to bind. In the second step, a wash step removes any unbound materials. And in the third phase or step, after substrate has been added, a colored product signal is generated and an intensity proportional to the amount of enzyme labeled analyte bound to the antibody is detected. The signal strength, as mentioned before, is usually inversely proportional to the analyte concentration. Non-competitive immunoassays, in contrast, first passively absorb to a solid phase, such as a microtiter plate, nitrocellulose membrane, or plastic beads. In a typical non-competitive immunoassay, antibody, often called the capture antibody, is present. Any patient antigen present would then be captured. Unknown patient antigen is then allowed to react with and be captured by the solid phase antibody. After washing to remove any unbound antigen, a second antibody with a label is then added to the reaction. Of note, these types of non-competitive immunoassays are more sensitive than the competitive immunoassays. In the non-competitive immunoassay format, the amount of label measured is directly proportional to the amount of patient antigen. In both competitive and non-competitive immunoassays, the label should not alter the reactivity of the molecule being tested, and it should also remain stable for the reagent shelf life. Different techniques using different types of labels are going to be discussed a little bit more in detail in a separate section of the chapter, but labels include substances such as radioactive substances, enzymes, fluorescent compounds, and chemiluminescent substances. In this figure, a non-competitive immunoassay is depicted. Here we have three examples. Sample A shows the negative control, in which no reactions take place. For sample B, immobilized reagent antibodies capture analyte in the patient sample, and these are denoted as the red dots. While in the antibody capture technique, in the last column, immobilized reagent antigen captures patient antibodies. The second step of the non-competitive immunoassay involves a wash to remove any unbound materials. In the third step, enzyme-labeled antibody that binds to different epitope on an analyte is added. So for sample B, it binds with the FC region, or on human immunoglobulin in the antibody capture step. A second wash step occurs after the addition of the enzyme-labeled antibody. Once a substrate is added and measured, a color change is observed. So the color intensity is directly related to the analyte, or human antibody, as depicted in the antibody capture method. The first type of immunoassay developed was radioimmunoassay. This method uses radioactive labels, as the name implies. The method is valuable in measuring a number of substances such as hormones, serum proteins, and vitamins that either occur at very low levels in blood plasma or are so small that they could not be detected. Radioactive elements have nuclei that decay spontaneously, emitting matter and energy. So there's several radioactive labels that have been used, but 125I has been the most popular, since it's easily incorporated into protein molecules and emits gamma radiation, which is detected then by a gamma counter. With this method, very low quantities of radioactivity can be easily measured. Although this method is extremely sensitive and precise, as you would imagine, the major disadvantage is the health hazard involved in working with radioactive substances. Disposal issues, short shelf life, and the need for expensive equipment has caused the lab to utilize other techniques for identifying analytes in low concentrations. With radioimmunoassays, the amount of label in the bound phase is indirectly proportional to the amount of patient antigen present. Enzyme immunoassays that use enzymes as labels were developed as an alternative to the radioimmunoassay. So the enzymes react with suitable substrates to produce breakdown products that may be chromogenic, fluorogenic, or luminescent. Examples of readily used enzymes include alkaline phosphatase and horseradish peroxidase, because these have the highest turnover or conversion of substrate rates, high sensitivity, and are easy to detect, so they're the most often used in various assays. With the enzyme immunoassay method, they're classified as either heterogeneous or homogeneous on the basis of whether a separation step is needed. The first enzyme immunoassays used were competitive assays, and these were based on similar principles of the radioimmunoassay. In this method, enzyme-labeled antigen competes with unlabeled patient antigen for a limited number of binding sites on an antibody molecule that are attached to a solid phase. After a washing step to remove any non-specifically bound antigen, enzyme activity is then determined. This method is usually used for measuring small antigens that are relatively pure, such as drugs and hormones. The tendency in the laboratory setting today is toward the use of non-competitive assays because they have higher sensitivity than the competitive method. Most non-competitive assays are either indirect immunoassays or indirect enzyme-linked immunosorbent assay, also known as ELISA. because the enzyme label reagent does not participate in the initial antigen-antibody binding reaction. The enzyme labeled reagent only binds after this initial reaction has taken place. This is one of the most frequently used immunoassays in the clinical laboratory because its sensitivity, specificity, simplicity, and low cost make it a desirable method. ELISA testing can be used to measure antibody production to infectious agents that are difficult to grow in culture or isolate, and for autoantibody testing. Some examples include HIV infection, hepatitis B and C, and then these enzyme immunoassays, ELISA techniques, can be easily applied to the point of care setting and also to home testing.
The steps involved in ELISA typically include an antigen bound to a solid phase, patient serum with unknown antibody, which is added and given time to react, a wash step in which an enzyme labeled antiglobulin or secondary antibody is added, and then a second antibody that reacts with any patient antibody that is bound to the solid phase. A variety of solid phase supports are used with ELISA method, including microtiter plates, nitrocellulose membranes, and magnetic latex or plastic beads. If no patient antibody is bound to the solid phase, the second labeled antibody will not be bound, and after a second wash step, the enzyme substrate is added. With ELISA methods, the amount of color, fluorescence, or luminescence is measured using a detection device and is compared with the amount of a product according to a standard curve. The last type of heterogeneous enzyme immunoassay discussed are capture assays. So if antibody rather than antigen is bound to the solid phase, these assays are often called sandwich immunoassays or capture assays. These are best suited to antigens that have multiple determinants. Capture assays are more commonly used for antibodies, cytokines, proteins, tumor markers, microorganisms, and in particular viruses, and they're also used in the measurement of immunoglobulins. Capture assay methods involve excess antibody attached to a solid phase that's allowed to combine with the test sample to capture any antigen present. After an incubation period, enzyme-labeled antibody is added. The second antibody recognizes a different epitope or binding site than the solid phase antibody and completes the sandwich. So depending on the particular enzyme used, either colored or chemiluminescent reaction product is detected. The enzymatic activity is directly proportional to the amount of antigen in the test sample. The next group of enzyme immunoassays are the homogeneous EIAs. So these ones are generally less sensitive than heterogeneous assays, but they're rapid, simple to perform, and adapt easily to automated methods. With homogeneous methods, there's no washing or separation steps needed. So their major use has been in determination of low molecular weight analytes such as hormones, therapeutic drugs, and drugs of abuse in both serum and urine specimens. Homogeneous enzyme immunoassays enzyme activity is directly in proportion to the concentration of patient antigen or hapten present in the test solution. And when antibody binds to specific determinant sites on the antigen, the active site on the enzyme is blocked, resulting in a measurable loss of activity. As mentioned before, homogeneous enzyme immunoassays are less sensitive than heterogeneous assays, and the sensitivity of these homogeneous assays is determined by detectability of enzymatic activity, the change in the activity when antibody binds to antigen, strength of the antibody binding, and susceptibility of the assay to interferences from endogenous enzyme activity, cross-reacting antigens, or enzyme inhibitors. Again, this technique is usually applied only to detection of small molecules, such as hormones, therapeutic drugs, and drugs of abuse. Rapid immunoassays are membrane-based assays that are easy to perform and they provide reproducible results. Although these are primarily designed for a point of care or home setting, many of them have been modified for increased sensitivity and can be semi-quantitative for use in the clinical laboratory. Typically, these are designed as single-use disposable assays in some type of plastic cartridge. They involve antigen or antibody being coupled to the membrane. The reaction that takes place is read by looking for a colored reaction. Some of these test devices require the separate addition of a patient sample, wash reagent, labeled antigen or antibody, and the substrate. Another type of rapid assay is called immunochromatography, and this combines all the previously mentioned steps into one. With rapid assay immunochromatography, the analyte is applied at one end of the strip and migrates towards the distal end where there is an absorbent pad to maintain a constant capillary flow rate. The labeling and detection zones are set between two ends, and as the sample is loaded, it reconstitutes the labeled antigen or antibody, and the two form a complex that migrates down, and this does so towards the detection zone. An antigen or antibody immobilized in the detection zone captures the immune complex and forms a colored line for a positive test, which may be in the form of a plus sign. Any excess labeled immunoreactant migrates to the absorbent pad. So in the figure, you have your clinical sample and your antibody to the particular analyte in question with colloidal gold. As the sample is applied, it flows down the capillary by capillary action, and then a test line appears in the first portion, and in the second portion is the internal control line, which indicates a positive test. So here, this is a scenario where the test is positive because there's a line seen at the patient test line and a line seen at the control line. This type of assay is used in various settings, for example, for pregnancy testing, also group A strep pyogenes in urgent care settings. So this is a very commonly utilized methodology. Fluorescent immunoassays are another type of labeled immunoassay in which antibodies could be labeled with molecules that fluoresce. These fluorescent compounds are all called fluorophores or fluorochrome. Immunofluorescent assays are restricted to qualitative observations involving the use of a fluorescent microscope. So what this means is that testing indicates if something is positive or negative or detected or not detected, so it's not quantitated. It involves grading the amount of fluorescence against a dark background. Immunofluorescent assays are used for rapid identification of organisms in cell culture or infected tissue, tumor-specific antigens on neoplastic tissue, and transplantation antigens. The two compounds most often used are fluorescein and rhodamine in these types of assays. Fluorescent staining can be categorized as either direct or indirect, depending on whether the original antibody has a fluorescent tag attached. In direct immunofluorescent assay, antibody that is conjugated with a fluorescent tag is added directly to the unknown antigen that is fixed to a microscope slide. After incubation and a wash step, the slide is read using a fluorescent microscope. Antigens are typically visualized as bright apple green or orange-yellow objects against a dark background. This technique demonstrates the presence of pathogens in patient samples. Examples of antigens detected by this direct method include Legionella pneumophila and Chlamydia trachomatis. Indirect immunofluorescent assays which are more commonly used than direct assays, involve two steps. So in the first step, patient serum is incubated with a known antigen attached to a solid phase. The slide is then washed and an anti-human immunoglobulin containing a fluorescent tag is added. This immunoglobulin then combines with the first antibody to form a sandwich, which localizes the fluorescence. These type of indirect immunofluorescent assays are used especially in antibody identification and have been the standard for detecting treponemal and antinuclear antibodies, as well as antibodies to a number of different viruses. These figures show a comparison of direct versus indirect immunofluorescent assays. So on the top figure, which is figure A in your textbook, this shows direct fluorescence in which patient antigen is fixed to a microscope slide and incubated directly with a fluorescent labeled antibody. The slide is then washed to remove unbound antibody. If specific antigen is present in the patient sample, fluorescence will be observed. Figure B in the below image is indirect fluorescence. In this method, specific antibody in patient serum, indicated in red as the patient antibody, binds to antigens on the slide. A wash step is performed and a labeled anti-human immunoglobulin is added, depicted as the blue antibodies in this figure. After a second wash step to remove any uncombined anti-immunoglobulin, fluorescence of the sample is determined. The amount of fluorescence is directly in proportion to the amount of patient antibody present. Of note, with these types of immunofluorescent assays, in general, they face the problem of subjectivity in the reading of the slides. Another fluorescent technique developed is fluorescence polarization immunoassay, which is based on the change in polarization of fluorescent light emitted from a labeled molecule when it's bound by antibody. If the labeled molecule is bound to antibody, the molecule emits an increased amount of polarized light. Incident light directed at the specimen is polarized with a lens or prism so that the waves are aligned in one plane. If a molecule is small and rotates quickly enough, the emitted light is unpolarized after it is excited by the polarized light. This method has been used to determine concentrations of therapeutic drugs and hormones. And of note, an example of fluorescence polarization immunoassay is developed by Abbott for amphetamine and methamphetamine in urine samples. With fluorescence polarization immunoassay, if sample analyte concentration is low, the antibody will bind most of the labeled analyte, as depicted here in the left image, restricting its rotation. When excited by polarized light, this emitted fluorescence will remain polarized, so the light waves will oscillate with the same orientation. On the right image, if a patient sample has high concentrations of analyte, this unlabeled analyte will occupy most of the antibody binding sites, leaving the labeled analyte free to rotate. The light waves emitted by the rotating labels will not be uniformly oriented, so in this example you would see less polarization. Fluorescence polarization immunoassay does require more sophisticated instrumentation, and it's the basis for several automated analyzers that are on the market today. The last type of immunoassay discussed in the chapter is chemiluminescent, and this is a technique employed to visualize antigen-antibody combinations. Chemiluminescence is the emission of light caused by a chemical reaction, typically an oxidation reaction, producing an excited molecule that decays back to its original state. Chemiluminescent immunoassays can be used for both heterogeneous and homogeneous assays, because labels can be attached to either antigen or antibody. A large number of molecules are capable of chemiluminescence, but some of the more common substances used include luminol, acrydium esters, ruthenium derivatives, and nitrophenol oxalates. So when these substances are oxidized, intermediates are produced that are of high energy state. These intermediates return to their original state, giving off energy in the form of light. The light emissions range from rapid flash of light to a more continuous glow that can last for hours. Depending on the substance used, different types of instrumentation are necessary for each kind of emission. Chemiluminescent technology is the basis for another broad array of several types of automated immunoassays, and these are used to detect a wide range of substances, including cardiac markers, hormones, vitamin D levels, and total IgE. So in summary, the major topics discussed in this chapter include that labeled immunoassays can measure small or low concentrations of antigens and antibodies, and labeling techniques can include radioactivity, enzymes, chemiluminescence, and fluorescence, with radioactivity being a technique not used as readily today in the clinical lab. There are two major types of immunoassays, competitive and non-competitive. And in competitive assays, all the reactants are added at the same time, and labeled antigen competes with patient antigen for a limited number of antibody binding sites. Non-competitive assays allow any antigen present to combine with an excess of antibody attached to a solid phase, and a second antibody bearing a label is added and binds wherever there is patient antigen. Immunoassays may be heterogeneous, in which they require a separation step, or homogeneous. Antibodies used must be specific and have a high affinity for the antigen in question. Radioimmunoassays based on competition between labeled and unlabeled antigen for a limited number of antibody binding sites. As mentioned before, in the non-competitive immunoassays, such as the indirect enzyme immunoassay ELISA, antigen is bound to a solid phase, an antibody is detected, and after binding occurs, a second enzyme-labeled antibody is added. In capture assays, the antibody is bound to the solid phase, and any patient antigen is allowed to bind or be captured, and a second labeled antibody is added that also binds to the patient antigen, making that sandwich. Fluorochromes are fluorescent compounds that absorb energy from an incident light source and convert that energy to light of a longer wavelength. Direct immunofluorescent assays involve antigen detection through a specific antibody that's labeled with a fluorescent tag. In indirect immunofluorescent assays, the original antibody is unlabeled. Incubation with antigen is followed by addition of a second fluorescent labeled anti-immunoglobulin that detects antigen-antibody complexes. Fluorescent polarization immunoassay is a type of homogeneous fluorescent immunoassay based on the principle that when antigen is bound to antibody, polarization of light increases. And finally, chemiluminescence is produced by certain compounds when they're oxidized and emit light as they return to their original ground state. One additional note when reviewing this chapter, please take a look at the study guide at the end of the chapter for labeled immunoassays. It provides a concise table of the types of assays discussed in the chapter, the basic principle, and the results that one would observe with the type of testing.